All right. Welcome, everyone, to the first virtual Pint of Science Gong Show in collaboration with Project Bridge. I'm really excited to try and do this. Obviously, with the coronavirus, we have not been able to do this in person. I'm really excited to have everyone. I'm really excited for our speakers. We have 5 really great speakers coming up, and we're going to talk about some of the rules. Everyone's going to get 3 minutes to talk. All the audience gets to make a bunch of noise, and it's going to be really cool. We're going to have some fun. Before we do that, I want to introduce Tebo. He's going to talk to us a little bit about Pint of Science in Norway and globally, and then we'll get into the show.
Hi, Matthew. Thank you. Welcome, everybody. I'm Thibaut. I'm the founder and director of the Pint of Science Norway. And I would like to actually introduce you the concept. So I will try to share my screen, if that works. It's always like on live demo are always a challenge. So let's try that. So can you see all full screen? Yes, seems like
we're good.
Yeah, okay. So Pint of Science is we are part of a bigger network, which is called Pint of Science. And this is actually a festival which is held every year, normally during the month of May. So during 3 days, we organize like events all over the world, so where researchers can share their latest discovery with you. So this is a perfect picture that will help you to understand. So it's a Pint of Science. We have banner, which is a bit our logo and mascot of Pint of Science. And it's a worldwide festival. On this festival, they take place in a relaxed place, so it's a pub of cafe, and they will never be able without the amazing work of volunteers. And the role is really to engage the science an exchange between the scientists and the general audience. So I would like to just go quickly through the different concepts that are around Pint of Science. So first of all, it's worldwide. So it's born in 2013, and it has been growing amazingly over the years. And for last year, for the 2019 edition, it was in 24 countries in almost 400 cities. So as mentioned, it's taking place into bar, cafe, where the general audience is really like relax and it's like to get in a chill atmosphere to really learn something about research and to have fun. That will be it will not be possible without the amazing speaker that's all over the world that doing amazing research and they want to share about it. On that, I think the main point, they want to share with you what they're doing because they want to show you how exciting science is, and they want to get your question as well, because at the end of the day, you are the main target for them. So the Pint of Science, we englobe every science. It has been clustered along 3 main themes. So we have Beautiful Mind that will cover like neuroscience and psychology. We have as well Planet Earth, which is we will speak about the field of earth science, plant science, and zoology. We have our society, where we will categorize like sociology, public policy, law, and history. Of course, we are interested by our body, so it will incorporate biology, medicine, health. We have Take Me Out, where we're going to speak about technology, engineering, computing. And finally, we have From Atom to Galaxy, which covers the field of physics, chemistry, and math. And everything will not be possible because we're all volunteers for Pint of Science. So we just give some of our time to organize this amazing festival, and that you can enjoy it. We can enjoy it, and the speaker as well can enjoy it. And you, as an audience, you can enjoy it as well by learning, but as well by having fun, doing some games that are organized everywhere during this festival. So as I mentioned, it's a worldwide festival. So we were 24 countries last year, and we continue to grow. So for example, this year, for 2020, it has been 6 countries, and Norway was part of it. So what does it mean, Pint of Science, for you, Norway? Sorry, I've got a slide. So this is the distribution of every countries that we have for the 2020 edition. So most of the countries are located in the old continent, but it's spreading everywhere. and we hope to see in the future, like in every countries in the world, so we can all learn about science in 3 days. And so Pint of Science for You, we were born last semester, and we are like 4 local chapters located in Tromsø, Trondheim, Oslo, and Bergen. And we manage, there's a central team, I'm the board chair, which we are located in Oslo. But our goal is to actually expand in every major city and to collaborate with the local players that are involved in science communication. So if you're interested, get in touch with us, and we will try to fund a new chapter with you. Thank you. So as I mentioned, our main event is the Pint of Science that was normally in May, but due to COVID, for now it has been delayed to September. We are still into the organization, see how we can organize or not. So please stay tuned. We will soon announce something on our social media. Back to you, Matthew.
All right. Well, thank you. So that's what Pint of Science is about. We're really excited to be able to do that again, have it in person across Norway, across the world. And Pint of Science works really well with Project Bridge, which is the organization that started this Science Gong Show. And like Pint of Science, Gong Show was founded in a bar. So if you want to take a minute and go grab a beer, so you can participate in that aspect of it, you are also welcome to do that. Project Bridge had a similar motivation to start. We were noticing that there were a lot of people who didn't really have a connection to scientists. So it's really easy when you hear someone say, well, scientists say we should do this. It's really easy to say, well, scientists are always saying this. They say we should have chocolate. They say we should have wine. They say we shouldn't have wine. But it's really easy. It's really a lot easier when you've actually met a scientist. So that way you can come up and say, oh no, I met this person. They're doing this cool research. We should really listen to them. So that's where the motivation for this came from. And so it's going to be really fun. We're going to have some presenters. And here's the rules. And this is where the audience participation comes in as well. So the speakers have 3 minutes to present. We'll have a timer. We will let them know when their time is counting down or when their time is up. But the really important part, and the important part as an audience, is that the speakers are not allowed to use jargon. As scientists, we have a lot of specialized words that we use that we understand that either have special meaning for us that's different than how it's commonly used, or they're just big words that we use because it's easier to say this one small word than it is to say, a bunch of long things, about it to describe it. So as an audience, your job is to catch the speakers when they use this jargon. And the way you do that, when we did it in person, was to have cowbells. We don't have that, so we're just going to make a noise. So this is an audience participation test. So if I'm going on and say, I used to study prosthetics. We use myoelectric. And when I use myoelectric, which is a word that, you don't hear very often. It's a specialized jargon word. So when I say myoelectric, I want everyone to type into the YouTube feed. It's over there, which is why I keep looking at. Type into it gong or rabble rabble or something like that. So we know that that's a word that we need to check and explain to you. So you can type the word as well. That'll help us understand because there is a little bit of delay. So let's try this again. This is audience participation time. If I say, I study myoelectric prosthetics, all right, audience, I need some responses from you. There we go. There's the front. So then I would have to stop and say, so myoelectric means that we pick up signals from the muscles, electrical signals from the muscles, and the computer inside the prosthesis translates that to motion. So that's how this is going to work. Oh, good. We got some audience participation going on here. So that's how it works. This is an audience participation thing. And this is also so that our speakers learn how to give better presentations. So I think we are ready to start. The first speaker we have is Petra, and she is going to be talking about how germ cells are real gems. So as soon as we figure, there we go. Hi, Petra. Tell us
Hey,
about how
everyone.
germ cells are real gems.
Hi. Yeah, so it's a great pleasure to open the very first year of a Norwegian Pint of Science. I'm a PhD student at the University of Bergen in Norway, and I study comparative developmental biology. Did you know you have immortal cells in your body? Cells that won't die with you if you give them a chance? Yeah, I'm talking about the gametes, so sperms and oocytes, that stand at the end of a line of one individual, but also form a beginning of a next individual, of its descendant. So sort of like a family line or a lineage that they indeed connect. And these cells also have a lineage, and it's called germ line. So it's formed by all the cells that form the end product, the gamete. So just like you can trace your ancestors in your family tree, we can trace the cells in theirs, and these are called the germ cells. So it has nothing to do with germs, and all the germaphobes out there don't have to pull out their hand sanitizers just yet. Before having a final form and gaining all the superpowers that are responsible for giving rise to brand new organisms, however, these cells have to undergo a complicated process called cell fate specification. And this usually happens in a development of an animal, where these cells have to accept or acquire a fate, which gives them properties that they have to use to fulfill a role in the body of an organism. And this is a unique trademark that we call molecular fingerprint. And there has to be something that makes these cells different from all other cells in a development, because they all start the same with a clean slate, let's say. So there has to be that decision of a fate. And I decided to look at how these fate decisions are made in diverse invertebrate animals, like a bootlace worm that can reach a body length of up to 50 meters, or a tiny microscopic marine worm that lives inside the gut of something called the red sausage, which is a Norwegian name for a sea cucumber that lives on the bed of the ocean bed or ocean floor, or a penis worm. And you may ask why we are interested in such a diverse and queer bunch of animals. And it's mainly because there are many similarities and differences in how animals decide these important fate decisions in their development. And it's only when we look at it as a whole broad picture, when we get a better view of how mechanisms work in the development of organisms and how they are either preserved or different, so convergent. And looking at how these animals are related to each other in the animal kingdom or animal tree of life, it can help us understand when did these cells, germ cells, appear in the history, and how the evolution sees the question of fate.
That is great. So thank you very much. I was trying to do a countdown on YouTube, and it cut me off. So I'm going to not do that. So that was wonderful. So yeah, it's really quick to give your talk in 3 minutes. Obviously, that was just an introduction. We've included people's Twitter feeds, so if people want to learn more about your work, they can go to your Twitter. Is that right?
Thank you. Yep, that's right. Yeah. That's Petra.
We have a minute if there's 1 or 2 questions. And in the future, if you have a question during the talk, you can ask it, and we can try and put it on the end. But if anyone wants to type in 1 question, we can see if we can present it. All right. So we got the first question on there. Why sea cucumbers?
Because they're super cool, because they spit out their gut as a self-defense, and because this animal that I'm interested in is not a sea cucumber itself, but a super tiny worm that is very flat and uses a sea cucumber as a house during a part of its life cycle. So they live as a symbionts, these marine worms that I meet in the sea cucumbers.
Nice. And then before I let you go and bring in the next speaker, what was it that initially got you interested in these?
In this research?
Yeah.
Just a passion for development of organisms and how different it can look, but also how similar the start point of a development can look and how it can generate different body forms that we find in nature.
Great. All right. Well, thank you. If people have any more questions, you can check out the that your Twitter handle there, and yeah, we'll have this up online to watch later if you want to review it or check it out again. So thank you very much.
Thanks again.
All right, audience. So that is what this looks like. I want to hear. There were a couple words in there that I wasn't quite sure about, so I want to hear a little bit more gonging from the audience. Don't hesitate. If you have a question about something, if you're not sure, just type it in and gong and ask your questions. So yeah, the next speaker we have is Charmaine. He is going to talk about the animal model of cafeteria diet, which is, I wanted to call animals in the cafeteria, but he wanted a more technical title. So we're going to have that. He's also our gong master, so might have to gong himself, but why don't you go ahead?
Well, I have Susanna that is going to go. So we are covering on that side.
Okay, great. Well, I'm going to start your timer and let's hear it.
Well, imagine the typical scientific laboratory, everything clean, everything under control. The smallest detail must be evaluated. And then I appear inside the lab with a package of hot dogs and muffins from the rats. Yes, exactly. I feed laboratory rodents with junk food. But of course, this is for a good reason. And in our research group, we are going to apply the cafeteria diet model. This consists precisely in feeding the laboratory rodents with the same industrially elaborated food, rich in sugar and fats, that make it highly tasty, but unhealthy. For example, potato chips or chocolate cookies. When I explain the cafeteria diet, people usually get surprised.
We got her going. So, Nancy would like to know about muffins from rats.
Yeah, this is the idea. If I finish it on time, you can get the answer. So yeah, the thing is that people like you get surprised about that, but the answer is pretty simple. We use cafeteria diet because it's a good model to mimic the current human eating pattern. The idea is that cafeteria diet is a translational model because we transfer a current social issue to basic research. But we need to go to the starting point. Everyone is aware that overweight and obesity are major health concerns worldwide, and its prevalence has increased dramatically in the last 30 years. There are many reasons for this severe increase. More or less, genetic factors explain 50%, and environmental ones the other 50%, making junk food a major actor. In science, the main method until now to induce obesity has been normal laboratory food with extra sugar or extra fat. Imagine the typical pet's food, but in this case for the laboratory. However, people didn't eat this sort of food. Among other reasons, people eat junk food because of its high palatability, because it combines sweet and salty tastes, and because junk food is varied and not boring. These features of junk food increase the amount of food intake, which is called overeating. Overeating could explain that 50% of the obesity problem. But just a minute, if it's not that simple that junk food is bad for our health, why people continue eating and eating junk food? Several research groups, including mine, starts to study the effects of junk food in the brain. And it's even possible that junk food could alter brain regions related to the process of enjoyable or rewarding stimuli, in this case, food. However, we still need to demonstrate that. But remember, eating is just not about nutrients, it's about enjoying. Therefore, the cafeteria diet is an excellent model for studying the effects of junk food on the brain, but also for a better understanding of the consequence of junk food on metabolism and obesity. And that's it. Thank you for listening to me.
Great. Thank you. That was great. So we can take an audience question or two, but I'm going to ask while people are writing. So how does your research stop me from eating a whole box of cookies?
What? Sorry, can you repeat?
Well, you were talking about these rewards and
Yeah.
how do you, so when I have a whole box of cookies,
Yeah.
does your research help me not just eat the whole box? Do you have any tricks for that?
Not yet. We already know as scientists that some foods creates the effect of overeating and it's sort of a snowball effect. With more junk food you eat, more junk food you eat in the future. But really, we don't know yet why our body is asking for more and more and more. For example, it's an hypothesis that is not proven yet. So it's just an hypothesis that maybe in the future it's demonstrated that junk food could have some sort of tolerance effect, like, for example, drugs addiction. But we need just to study the effects on the brain to reach this conclusion.
Okay. We have an audience question. Dorothy would like to know if the cafeteria diet is a model for a junk food diet.
Yeah, exactly. This is a problem here in Norway because usually cafeterias could be associated with healthy food, but in other places, this model is called cafeteria diet model. I don't know really why it's called not it's called junk food, that it's more logical name.
All right. Well, great. Thank you for your presentation. If anyone has any questions, you can go to your Twitter, and which was in the the bar for a minute. Oh wait, one more question. How do you measure the brain's reaction to muffins and other cafeteria food?
Wow, this is a huge question. There are different ways to measure that. We can, for example, take samples from the brain and analyze, for example, the amount of the expression of one neurotransmitter, like the one from dopamine. We can, for example, try to use in vivo techniques like fiber photometry or optogenetics. So yeah, this is more or less the two main techniques. But yeah, we need maybe another edition of the Gong Show just to talk about one of them.
All right. Well, great. Yeah, thank you. All right. So audience, I've seen a couple of questions. I've seen a couple of gongs, but this is the Gong Show. This is audience participation. So I'm not asking you to be mean to our next presenter. She's got some really cool stuff going on. I'm just saying, use those gongs. I miss hearing the cowbells. So our next speaker is Susanna. She is going to talk about brain and movement in long living. And we're going to
Okay. I will talk about brain and movement in long living. I use in the title this long living term instead of aging because I wanted to emphasize that aging is a process that is shaped throughout life by the experiences, habits, and the environments surround us. The more we live, the more factors influencing the way our brain, our thoughts, and our movements are. The brain is especially sensitive to the food we eat, the number of hours we sleep, the time of type and amount of physical activity we do, the intellectual activity we perform, the substances we take, like coffee, alcohol, or others. And the effects of all of these factors are also modulated by our genes. As a result, our brains work differently in different people and at different ages. These differences are reflected on the way we think, speak, and move. When we talk about physical activity, it is commonly seen as different from thinking. But the connection between movement and thought is more evident. For example, if we think of a complex movement, we need to pay more attention to it. The mental processes that control movement are called psychomotor functions. These psychomotor functions control gross movements, like when we kick a ball, or fine movements, like when we draw a figure. During aging, movements and thought normally become slower. But how slow isn't normal for a senior? Are slowing and lack of precision in movements the consequence of a disease that is being masked by aging? How are the differences in movement between healthy seniors and those with a degenerative disease reflected on the brain? Well, all these questions are the directives of my research in aging neuroscience. One interesting fact is that the brain is not completely symmetric. So, like, we use our hands differently depending on the task. For example, a left-handed person will use more the left hand to draw, more to pick some object, and the right hand maybe to other activities that require less precision. Our brain is also used more for, our left hemisphere is used more for some activities, and the right brain is used for other activities. Depending on how much we use certain regions of the brain, the connections of these regions may be strengthened or weakened, and the size of these regions may increase or decrease. Our recent data in older adults indicate that seniors with mild deterioration in the intellectual ability tend to lose asymmetry in their movements. And also this loss of asymmetry in the movements is associated to loss of asymmetry in the integrity of the connections. So do more exercise and try to do new exercise to learn new things, new movements, and new activity with your brain. Thank you.
Great. Thank you.
Okay.
Yeah. So that's exciting. So this research is about how to stay active and functional as we age. Is that correct?
More like there's one important thing, the activity we do. So there are two aspects. One is studying how activity already is, how movements are already, for example, in an older adult, because it's not only, it is slower. We know, we think of an older person like a slower person,
Mm-hmm.
but how slower the person is the person sick or just old? It's not the same. And the other aspect is what you are mentioning, the type of exercise a person can do to be better, to have a better brain health, not only physical health, but brain health. And it's the activity that is not structured, like walking, like taking care of grandchildren, like going to the park, going with friends, cleaning the house. That is the kind of activity that makes the difference of people with slow activity in the brain or normal activity in the brain.
Nice. All right. So we had a couple of questions. The first, a couple of people asked about what you just said. What kind of activity is good for the elderly? I want to ask. Dorothy would like to know: can you teach an old dog new tricks?
Yes.
Okay.
Yes, the plain and simple answer is yes. It's not as easy, probably. It also you also need to convince the dog, but it is possible. Yeah.
Okay. And then the next question I want to ask is: does activity in younger years help when getting older, or is the current activity more important? That is, I think the activity as you're older.
Both are important. So the activity in younger years help very much. You have some kind of reserve, and that's good. But of course, if you just say, "Oh, I do have reserve," and don't care and stop doing exercise, then it can even be worse.
Okay.
So
I did
a long life thing.
Okay. And I did say that was the last question, but my mom asked this one, so I'm going to ask it. What about exercising the brain and learning new skills like crosswords or bridge? Is that part of your study?
We are not studying that yet,
Okay.
but we know that it's important, especially But other studies report is that it is very good to learn new languages, and also learning things like dancing, like taking classes, learning some new dance.
Great. All right. Well, thank you so much for presenting. If people are interested, let me pull that back up. They can check out your Twitter, and this will be available online later as well. So our next speaker is Thibault. Our co-host, our co-founder, is going to talk to us about boosting drug discovery with computers. Although he has to add himself to the stream. Yeah, there we go.
I'm the tech guy, so I have to do everything myself.
All right, so he's the co-founder, the co-host here. So definitely don't hold back your gongs on this one.
Okay, thank Matthew. So actually, I will present to you the research I've been doing when I was taking my PhD at the University of Tromsø. So I will start. So do you know that I could actually trade that to you for 12 years of labor and 20 billion Norwegian NOK? Thanks, Matthew. Because this is actually the average cost that it takes to bring a new drug to the market. And for one success, actually, we have 100 that fails. So what can we do to get more drugs on the market, cheaper and faster? Well, actually, I might have the start of an answer. This is my tool I use for drug discovery. This is the computer that I perform computer-aided drug discovery, or CAD for short. So it's a field of research when we use computational techniques to find new medicine, new drugs. And it comes with a lot of advantages. For example, it allows us to explore a bigger set of data, a bigger volume. It is estimated that in 2030, it will cut down the cost by 30% for drug discovery, and we can transform years of research to months. And this is the research, this is the approach I used for my PhD research. So we wanted to find new treatment for brain disorders, especially depression, because if you take medication for depression, well, you have 1 chance out of 3 to not respond to the treatment. And if you do respond, it comes with a long side effect that nobody wants. So there is need of better drugs or better treatment. So my research group identifying a new drug target. So when you take a pill, the molecule need to interact with the biological target in the body, that is referred as drug target. My research group identifying a new drug target that might be involved in depression, and we wanted to find a drug that will interact specifically for this drug target. So we assembled virtually a database of 8 million small molecules, and by applying several techniques of computational techniques, we filter down this number of molecules until a selection of 60. That was the 60 most promising, because these computational techniques, they can only make prediction. So we bought these 60 compounds and we went to the wet lab, and we found that among these 60, 5 of them were active against this new drug target. So among the 8 million, we found 5 at least that were active. So this technique, I'm not here to replace. Okay, I will set up. So next time you take your treatment, be careful. Right. The treatment might come from my computer.
All right. One more, yeah. You want to just wrap that up?
Okay.
All right. Anyone? Okay, so we have a couple of questions for you. The first one, and this is something that I'm always wondering about, is where do you obtain the data to do those studies?
So actually, you go in the scientific literature, and there's as well some databases that are publicly available. And of course, the big pharma or some have their own database that are private, and so we don't have like access unless you work with them or you are at the company. And some allow virtually, because there is different kind of molecule that we have to, as well as Charmaine, I need to do another one to explain everything about the data.
Okay. Cool. And can people do this from home, or is this something that's like only for researchers?
No, actually, everybody can do it from home. You can just rent some CPU in the cloud from Amazon, from Google, from Microsoft, and you can do it yourself. You just need to have the proper tool. So you pay for either for licensed software or you can find open source software. But if you have the knowledge, you can find like clone computing for cheap nowadays.
Cool. And have you, what diseases are kind of together? What diseases have you done this for, and is this already being used?
So we tried actually for, as I mentioned, to treat depression, but this receptor that is involved in many mental disorders, and it's not yet used. I mean, it's still in the lab working. There's still some experiments, but I don't know if that will go to the markets. I mean, the tool just increase the chance to go to the market, but it's not the final solution. It's just a help.
Right. And I know there's several labs in Norway working on stuff like this, and I'm sure elsewhere in the world working on using computers to speed up this process. The last question is, did you design the computer program for it, and like how does that work, and is it is it efficient?
So there's already like, I didn't design any software. I'm just using the software. So you can make prediction. So if to confirm, so you can make some prediction to see if the drug will work or not. So then you get the best one. There is a score, and then you need to go into the wet lab to confirm what you see on the computer. We just make prediction. They say it's like just prediction. Everything done in the black, it's not a black box, but in the computer is actually a prediction.
Great. All right. Well, thank you so much. If anyone is interested in getting some more information, that's your Twitter handle. And we are going to move to our next speaker. We have two more speakers, really excited about them. I'm mostly done my beer, but if anyone else wants to go. This is usually the point in our presentation where we tell people to go to the bar and get another beer, but everyone's at home, so yeah, there you go. Dorothy is going to talk about responsible research and innovation in marine science. And for those of you who are not in Norway or maybe in the EU, responsible research and innovation is a sort of guiding principle, right? For that governs really a lot of the research going on here, sort of a code of ethics. So it's really exciting to hear someone talk about this. So Dorothy, you want to go ahead and tell us about responsible research and innovation in marine science?
Yes, thank you, Matthew. So if there's one thing that Corona has taught us, is that a little thing could go a long way. You can think of like the snowball effect. As the snowball goes down, it gets bigger and bigger. And also the butterfly effect, that the flutter of a butterfly's wings in Brazil can cause a tornado in Texas. Everything in this world is interconnected, and the corona pandemic has really shown us why we need to be prepared for these interdependencies in our world. Greta Thunberg, the Swedish high school student, also has showed us how one voice can actually spark a revolution of a view. The younger's view that many young people today understand that the decisions we make about climate change today will affect our actual climate tomorrow, and why this needs to be put on the agenda. But one of my main research questions is, how do we prepare ourselves for climate-induced snowball effects or climate-induced butterfly effects? What I did was put together a radically interdisciplinary team together to look at transdisciplinary methods. Now, by this I mean the radically interdisciplinary team is different than just the biochemistry team, which is focused on the natural sciences. Radical interdisciplinarity means natural science connected with social science and the humanities. And the transdisciplinary aspect is, how do we go from inter to trans? We have to leave the university, leave academia, and engage with society. So this is the transdisciplinary aspect. So what we did was connect ocean models with fish distribution models, where the fish are located in the ocean, with IPCC worst case climate scenarios. Because we know that worst case scenarios, especially the worst case scenarios, will have effects on our international relations, on our economic situation, and our jobs and culture, really, just how Corona has showed us that. So what we did is create these scenarios of the future, these plausible scenarios, because we know that climate change will affect international relations. One thing that could happen in Norway is that because of climate change, the cod, the Northeast Arctic cod that we're very dependent on, could go totally into the Russian zone. What would happen if the Russians told the Norwegians that they're not allowed to fish cod anymore? What we did was that we wanted to see the generational effect of this scenario. So we asked high schoolers, their parents, and their grandparents what they would do in this situation, combined it all, and talk about the importance of intergenerational climate democratic deliberations. That is responsible research and innovation in marine science, as I have interpreted recently.
Perfect. I think that was the closest we've gotten to exactly 3 minutes.
I guess practice works.
Oh, of course. I really like that you're bringing in different, not just other aspects of the sciences, but humanities and social sciences. I feel, and I think a lot of us feel that science is wonderful. It's great on its own, but it really needs to be shared with the community, shared with people, and brought in other aspects like social aspects and economic aspects and humanities aspects. So it's really great to hear that you are working on that. I don't see any questions yet, but I did see just towards the end I saw a gong for the IPTC. Can you say?
IPCC. The Intergovernmental Panel on Climate Change. So this is UN's special group I guess the IPCC has been in existence for a couple decades now. Somebody on the internet can probably Google it and correct me. And they won the Nobel Peace Prize they shared with Al Gore. When was that? Maybe 6 or 7 years ago. So this is the group. I mean, I think there's hundreds, if not over 1,000 scientists all over the world that are collating data, interpreting data on climate change, and they have these regular reports that come out.
Great. And then and then we have one other question. How do you increase the interdisciplinary work or make it easier? It's a really important thing to be doing interdisciplinary, cross-disciplinary work. How do you do it?
That's a really good question. I think a simple and kind of playful answer is to tell stories. Stories usually have a protagonist, a conflict, and you have to figure something out. And for example, when we tell the story from the perspective of a concerned 18-year-old or a concerned 50-year-old fisherman or a policymaker that has very complex situations, we understand that the answer isn't found in the book. The answer takes these different aspects of nature and of society, and we kind of have to work together to put these different pieces of the puzzle together.
Cool. Great. And then have one other question. How do you follow up on the findings? Like, do you make policy because of this? Like, what comes from this?
This is a really good question. And so what I just described, these intergenerational dialogues, is a paper that has been accepted in Frontiers in Marine Science. So I'll be tweeting the paper to everyone. And one of the conclusions that we make is that, like I said, there is no answer. There's no one. The answer is not 42 in the situation on how to be prepared. But we feel that democratic deliberation, so the act of discussing these things, not just in our heads or in chat groups, but verbally discussing this from a local perspective to get local governance ready to, how could we be ready for something that hasn't even come on the agenda yet? So we find that getting these different generations together, that helps us be prepared for when something does strike, that we've had interesting discussions and we're ready to pounce on this and find collaborative answers in a much shorter amount of time than it would take. It also creates trust with local governance and and citizens as well.
Great, thank you. So let me put your, so any other questions or follow-up? There's your Twitter handle. We're going to go to our last speaker. I'm really excited about this. So Erin Golden is the founder of Project Bridge in Colorado, and it's earlier in the day there. I guess it might be a little early for a beer, but Erin and I developed this Science Gong Show together and had it in bars in Colorado, and so I'm really excited that she's able to come and talk to us. Give her presentation, and her talk is called "Can You Hear Me Now?"
Can you guys hear me now? Seriously, is my microphone working? So hi everybody from Denver, Colorado. It is 10:30 in the morning here, so I'm drinking my coffee, not beer. So I am Erin Golden. As Matt said, we started the Gong Show together, so I'm really excited to be doing this internationally. I actually don't work in the lab anymore. I am now the Director of Undergraduate Research and Creative Activities at CU Denver. So basically, I like doing Gong Show so much, I decided to go teach undergrads to talk broadly about their research too. But when I was in the lab, I worked on sensory cells. So the cells that allow you to interact with the world around you, and understanding why some of these cells can regrow and others don't. So if you guys think about when you take a sip of a hot coffee, like what I'm drinking, and you burn your tongue, it's kind of the worst, right? For the rest of the day, your tongue feels weird, and food doesn't taste quite right, and then it's just basically annoying. But luckily, within the next day or so, your tongue heals, and you go on with your life as normal. And that's because the cells that are on your tongue that produce both your taste buds and the tongue skin that surrounds them is really good at regrowing new cells. And my research looked at how these tongue stem cells know when to make a new taste cell and when to make a non-tasting skin cell. No, this is not Irish coffee. So yeah, looking at when tongue stem cells can know when to make a taste cell versus a non-tasting tongue skin cell. So there's a lot of reasons why this decision is important, but the reason why I was particularly interested is because it turns out that not all sensory systems have a stem cell population that can do this very well. So if you take, for example, hearing, within your inner ear, there are specialized cells that process sound waves into a language that your brain can actually understand. And when these cells are damaged, they don't grow back, which is why nearly half of Americans over age 65 have some form of hearing impairment. And this is also why you should always wear ear protection when you go to loud concerts. It turns out that the ingredients that your body uses to repair tongue cells are also present within your hearing cells. They're just used in different ways. So you can think of this like how a piece of chicken, a tomato, and an onion could be used to make an Italian food dish or an Indian food dish or a Mexican food dish. They're just used in different ways and with slightly different processes and accompanying ingredients. So my goal as a researcher was to understand how the body uses different genetic ingredients to make, say, a taste cell versus a hearing cell, with the hope that this understanding would someday give us the ability to regrow hearing cells like we regrow taste cells. All right, and I think I'm done. So with my couple extra seconds, how much?
20.
20. All right, fun fact. Non-mammalian vertebrates actually can regrow their hearing cells, but mammalian vertebrates cannot. And that's because they think this ability was lost when we developed a coiled cochlea, so inner ear, that allows us to have high frequency hearing. All right.
Cool. All right, we have one question about taste. My Indian friend lost her sensitiveness to hot food when she moved to Norway. Is that normal? And just for some context, in Norway, they don't seem to really do spicy food. It's been hard to find spicy food here. So is that normal? Is that because of the food? What do you think is going on, maybe?
So spicy actually isn't a taste, first of all. Spicy is sensed by the same neurons, free neurons and fibers in your skin and tongue that allow you to feel temperature and pain, which is why you sweat when you have too much hot food and too much pain. These neurons, these free nerve endings that sense hot food, can adapt over time. So you get less sensitive to spicy the more you eat it, because your body has mechanisms to kind of just become less sensitive. It's like, you don't notice that you're wearing your underwear all the time because your body knows how to like turn off the signal of your underwear being on your body all the time. It'd be annoying if you were constantly noticing that. And so, yeah, if you stop eating spicy food, you can get less sensitive to it or more sensitive to it. I also found after I was pregnant, after I had a child, I was really sensitive to spicy food, and I used to eat tons of spicy food. So. Body does all sorts of crazy things.
All right. Well, cool.
Somebody asked, after becoming hearing cells, how do they connect to the rest of the brain? So yeah, the inner ear is super beautiful, and you have hearing cells.
I'm fighting over trying to show this question.
What is happening to you? Too much beer.
Go ahead. How do they connect to the rest of the brain?
Yeah, so your hearing cells in your inner ear are arranged in these beautiful rows and patterns, and you have neurons, which are the cells that connect all of the different sensory cells in your body to your brain, running along the length of the cochlea, which is the inner ear. And so basically you can think about it like electrical wire. You have these long electrical wires that actually attach to the cells in the inner ear and then connect them back to the part of the brain that processes hearing and sound.
Cool. All right. Well, thank you so much for your presentation. Thanks for joining us all the way from the other side of the world.
It's pretty cool to be doing this.
Yeah. All right. So everyone, that is our presentation. That is the virtual Science Gong Show. I put up at the bottom here the Twitter handles of all of our speakers. Do check them out. Do check us out. See what we've been sharing. See all the other work we've been doing. Pint of Science should be presenting in September this year. We're still not sure if we're going to do it online or in person, but we also hope to have some more of these virtual Science Gong shows, maybe with some more gonging, maybe with some more beer, with a new set of speakers coming up in the next month or two as well. So I hope you had a great time. Feel free to contact us if you have any questions or if you'd like to participate in the next either Science Gong Show or Pint of Science. And we will see you next time. Thanks.